All right, we will now go to our last speaker, Konstantinos Petousakis, who's a PhD candidate in the Parazi lab at IMBB 4th. Probably mangling that name again. So in this talk, we're going to hear about an expansion beyond the neural point model, looking into how you could consider different dendritic domains differently in computations, because neurons are very complicated and difficult to convey from like a single point. So please take it away, Kostas.
Very much, Emily. Unfortunately, no interneurons here. Can everyone hear me? Give me a verification, please.
Yes, we can hear you.
Okay, so hello from Greece, everyone. Today I'll be presenting this work on the contribution of apical and basal dendrites on orientation selectivity using computational modeling. Some quick basics first. It has been known since the 60s that the presentation of stimuli of different orientations produces different responses in a V1 pyramidal neuron. Orientation selectivity is the phenomenon that is exhibited by neurons whose response is a function of input orientation and is usually demonstrated by the orientation tuning curve. Orientation selectivity is an input-dependent phenomenon, and it has been shown that V1 neurons receive both feed-forward visual input and feedback attentional and predictive inputs. And these inputs primarily reach the basal and apical trees, respectively, as you can see here. In addition, the synapses of V1 neurons themselves exhibit orientation or direction preference and can exhibit different preferences even when situated on the same dendritic branch. Next, an introduction to the model neuron we used, which is a morphologically detailed and biophysically accurate layer 2-3 V1 pyramidal neuron. And akin to real neurons, the model neuron also features per synapse orientation selectivity, with synapses varying in preference even when situated on the same dendrite, as you can see in these polar plots. Our first step is to characterize the nonlinear behavior of a dendrite, which we do by evaluating the spiking thresholds for sodium and NMDA spikes. And we also quantify the extent of dendritic nonlinear behavior through the NRLE metric, which you can see here, and is the max ratio of actual to linear extrapolated output for each dendrite. What we find is that all dendrites exhibit a wide array of different nonlinear behaviors. And naturally, we also verify that our neuron can exhibit orientation selectivity, which it does. And orientation tuning persists even when the input is not evenly shared between the two trees, which is the biased condition, the red curve. To further investigate the role of dendritic activity, we inspected samples of contemporaneous somatic and dendritic spikes, noted that the dendritic spikes tended to temporarily precede somatic spikes, as you can see on this detail. The red trace precedes the blue trace. If the model is accurate, then the proportion of somatic events preceded by basal events should be comparable between the model and in vivo two-photon data. You can see a sample here. To verify this, we converted model voltage recordings into two-photon-like traces using a combination of Gaussian filtering and downsampling. The resulting traces were processed in the same manner as raw GCAMP6 fluorescence data. Both the model and experimental data then pass through an event detection pipeline, which characterizes the timing of somatic events as dendrite first or soma first. You can see samples here. And comparing the results from both datasets, we can see that they are indeed comparable when looking at the fraction of dendrite first events, which is 49% for the model and 45% for the experimental. That's the percentage of overall events. Finally, we attempt to ascertain how dendritic spikes contribute to somatic spike generation in a causal manner. To do that, we record a large number of spikes and rerun identical simulations in which either the apical or basal sodium channels are blocked right before a non-somatic spiking event. And the result is either the survival or extinction of the somatic spike we're trying to characterize. And this allows us to label all somatic spikes as either apically-driven, basally driven, unstable, or bistable. And we find that the majority of somatic spikes appear to be driven by the apical tree, which is counterintuitive because basal dendrites are the ones that receive the vast majority of actual visual input. However, simulations in which we remove all basal stimulus-driven input fail to generate any somatic spike, which obviously indicates that the basal tree has a key role, which we already know. To identify the true cause of the somatic spikes, we fed specifically selected features of this spiking data to a machine learning classifier, training it with independent spiking data, which means means data where only one dendritic tree exhibited spikes before the somatic spike, or cooperative spiking data, which is data where both trees exhibited spiking before the somatic spike. The performance of the classifier was greatly enhanced when training with the cooperative spiking data, suggesting that the mere combination of independent spiking samples did not suffice to generate the same amount of information as cooperative spiking, which is evidence for the existence of a synergistic effect between the apical and the basal tree. In conclusion, we have used a detailed computational model of a layer 2,3v1 pyramidal neuron in order to discover how apical and basal tree activity shapes neuronal output. We find that although most somatic spikes appear to be causally instigated by apical dendritic spiking, a subtle intra-tree dendritic cooperativity effect is the true culprit. And we hypothesize that the basal tree generates a backdrop of depolarizations and dendritic spikes, which are shaped by visual input. Meanwhile, the apical tree intermittently produces dendritic spikes due to predictive and attentional inputs. And whenever an apical dendritic spike spatiotemporally coincides with a sufficiently large basal depolarization or a spike, a somatic spike is generated. And this could be a means of attention and prediction-based feature extraction for visual input. And this concludes the first part. Thank you for your attention, and I'll be glad to take any questions.
Thank you so much. That lovely talk. I have a kind of a broad question. Hopefully that I'm not asking a dumb question. This is going a little out of my like realm of understanding. But I would love to have one question that I have while we wait for our audience members to ask questions. And then if you have any other slides that you wanted to talk about, I'd love to hear those as well. So my kind of broad question was, right in the discussion, you were talking about this maybe cooperativity between basal and apical spikes, where the basal spikes are much rarer, but seem to be necessary in order to generate a soma spike. And you're talking about like feature selection. So what do you think might be the actual like circuit mechanism to coordinate these apical and basal spikes?
It is hard to tell, but what this essentially boils down to is a form of predictive coding, where the entire layer essentially is being guided by the layer above it, and perhaps even higher order cortices or areas of the brain, and essentially synchronizes so as to predict the actual input itself. And whenever there is a mismatch between the input and the output of this particular cell or network or layer, there is an error signal that propagates to upstream areas, which then eventually returns and tells to the network, okay, you made a mistake. This is not what we expected to see. I'm not sure if I've completely covered your question, but the overall network level mechanism is essentially a predictive coding model. I hope that catches all of it. Yeah,
that answers my question. I'm curious if you have any more. You said this is the beginning of the talk. Did you have even more stuff to share with us?
I do have a little bit more, yes. I mentioned the different profiles of nonlinear behavior of the dendrites. You can see those here. You can see how wide the gamut is of the responses for each dendrite. There is even a single errant sublinear apical dendrite right here. And you might be wondering, well, okay, so why do we just check the dendrites themselves and what they do if all we're checking is orientation selectivity? And the answer is that it is pretty difficult to actually ascertain what's going on when just looking at orientation preference. We checked for this by changing the mean orientation preference of each tree so that we introduce a level of disparity between them. So each tree has a different mean orientation preference. When we do this and we run the experiments, we see that the overall preference of the neurons seems to be around what you would expect if both trees contributed equally. So we can't really make heads or tails of this kind of result, which led us to consider the contribution of dendrites in a more detailed manner. And so we turn to what exactly happens when the dendrites produce sodium and NMDA spikes, and then that moves on to the soma.
Very cool. We have a question. We have a great question from Jason Moore, who asks, if not all apical events propagate to the soma, would this predict higher activity rates in the apical dendrite compared to the soma?
What happens in the particular model, I can't make an inference about every neuron, but in this particular model, you'll note that we do have an apical trunk, and we have a very extensive apical tree, which mainly features short and thin distal branches, which are extremely excitable and easily produce sodium spikes. These then move on to the next compartments where the sodium channels also generate new sodium spikes, and this chain moves on to the apical trunk where the overall signal is heavily amplified and easily reaches the soma. This is not the case for the basal dendrites, which, as you can see, sprout from the soma independently. There is a single branching point, and so this synergy of the branches themselves is not seen on the basal tree. So we do see most apically generated spikes reaching the soma, but that is not the case for the basally generated spikes, which, due to the current sink effect, the spikes usually just become heavy or medium-sized depolarizations. That's not to say that no basal spike propagates to the soma, but most don't actually make it there.
Okay, Jason Moore had a follow-up question to that, which is this observed experimentally?
The problem with observing these things experimentally is that it's very difficult to do patch clamping on specific dendritic branches and keep track of them for a sufficiently long amount of time. I think that currently there are some labs that are doing such experiments, but no such data has unfortunately found its way to us so that we can verify it. But I would be extremely excited to get my hands on data like that.
I imagine that's it's true for a lot of modelers.
Yes,
well.
I'm sure.
Yeah. Does anybody else from our previous speakers have any questions?
Yeah. So if you were really to simplify this this model, how would you combine this bit of binary logics with the firing rate model for a neuron?
That's a tough question. The simple answer is I am not really sure how I would combine this. This is definitely not something I can just answer in a single minute, but the direction I would probably try and take this in is some sort of filtering, because as I said, this essentially is a kind of feature extraction. So what you would have to do would be to implement some way of reducing a very high dimensional input, which is essentially what reaches the basal tree, into something that can actually be coded by a finite population of neurons and synapses. Visual input is obviously infinitely complex, and not all of it can be represented in the brain. There has to be some sort of mechanism that shrinks it down and fits it into the finite population of neurons and synapses that we have. And what you would have to do is essentially implement some sort of dimensionality reduction. I am not exactly sure how you would do that. There are many ways, obviously, but the exact implementation now eludes me. I can't be sure.
Okay, thank you.